Well, welcome to part 3 of the IDA Consultants Regulation of Advanced Therapy Medicinal Products, or ATMPs, recorded in February 2010. In part 1 we talked about the regulatory framework for developing these products in Europe and how important it was for not only European companies but those in the US looking to raise investment and explore markets. In part B we talked a lot about the clinical trials regulations and things like that, and we started on the preclinical safety for cell-based systems. There's such a lot of material here that we're going to part 3, so this is part C of the video. We may even end up in part D, but we'll see how we get on. There's such a lot of information here. Again, mind maps, get those mind maps, so important. ATMP at damienbove.com, and anyway, without further ado, let's get back to the topic in hand. So we were discussing the toxicology requirements, and I was flying through at lightning speed before we decided that actually we needed another video. And we talked about the design of your toxicology studies, the duration being the same as the clinical programme, but also including a huge recovery time to incorporate the time that the people are going to be exposed to your technology. It could be in there for a long time. But there's also proof of concept data that needs to be conducted as well for a package, and that can be the cell fate. You put the cells into the body, where's it going? So this is your traditional ADME, so your absorption, distribution, metabolism, elimination. But you need to know where those cells are going, how long they're hanging around for, and how the body's getting rid of them. You need to be looking at your pharmacodynamics and your safety data. Again, duration is so important. Biomarkers, what are they, how are you going to track them, and adverse events as well. And species, again, all these things. Species is so important. So is route of administration, as close to your clinical as possible. Your dose response data is going to be absolutely critical in developing your toxicology and your risk management and your clinical programs. And again, formulation. Before you start your tox, make sure that formulation works, because it's got to be what you use in the clinic. It's going to be difficult for you to use the data if it's not the same, so be really careful with that. You need to look at the source and origin of your cells. Are they autologous? Are they going back to the donor? Are they allergenic? Are they different from a different person? Are they xenogenic? Are they from a different animal? And this is all really important in the risk management of these processes. And is it a combined product, cells, device, etc.? And is it genetically modified? I mean, all these things are going to be really important, and they need to be incorporated into your planning and designing. As I talked about, species selection is always case by case. They've got to have the relevant pharmacology. You can use non-standard animals, so you can use genetically modified animals for your proof of concept. That's very acceptable. And the species selection is can limit the size of your preclinical. If there's just not available relevant species that's going to be pharmacologically relevant, don't do 2 species. It's a waste of animals. It's unethical. Or you can establish no adverse event level in one species, do a single dose in another to confirm that in 2 species. If you can confirm that in 2 species, that's enough. So you're looking at one program and then a confirmation study on 1 dose. But again, it's always case by case. You need to take advice. You need to speak to the professionals. So go to my website, damienbove.com, fill in the contact details. I can help you, or I can put you in touch with people that can help you. Take advice early. That's all we can say on that. This brings us on to the preclinical for gene therapy. Again, this is all case by case. There's no standard packages available off the shelf. You need to take advice. I mean, it's so important. We're talking transgenic products. What's the effect on the immune system? Are they genetically modified? What are the requirements to be shown? You need to have your in vivo and in vitro activity. You need to understand that. You need to understand the biodistribution of your technology. You need to understand the target tissue specificity. You need to be looking out for unintended effects, and you also need to be doing integration studies. How does your gene therapy integrate into this? And if it goes anywhere near the germline, that's it. You're back to the drawing board. In Europe, anyway. You need to be looking at the applications of your technology. Is it ex vivo manipulation? Is it in vivo administration of gene products? Are you inserting a function? Are you regulating transcription? These are all things that are going to impact on the design of your preclinical program. DNA vaccines, they're not considered gene therapy, but if it's combined with an immunomodulator, then it is considered gene therapy in an ATMP, so you have to consider that. The delivery of your product, again, needs to be as close as possible to you and you envisage your clinical use. The vectors that you're using, what do you know about the vector? Where's it been used before? How safe is that? What's the background? Data, get all this information. Gene products can have cascade effects, so you interfere with a particular function. How does that function then cascade through the rest of the body? This is all important. You need to understand this so you can look for it in your preclinical safety. How's the immune system going to respond to these changed products? Is there a pre-existing immunity in an animal against the vector, for instance? That can be really important. And so get existing data, look for your vector, look for the transgene or the recombinant work that's been done by other people in the area. Look at the literature on the gene, really important. You need to fully understand that and select your animal models. What's been used in the past, what's been used in your discovery research, because that could be the only animal model you can use. We touched on integration, so you need in vivo data and in vitro data on that. And if you, like I said, if you get germline integration, that's project stop. It's unacceptable to any of the regulations in the world that we know of. You're not going to get anything onto market in that state at the moment. It's just too much of an unknown. So you need to be looking for that, and you need to be looking very early for that because it's important. Again, viral vectors are very popular with these technologies. What's the inflammatory effect of that viral vector? What's the opportunity for mutagenesis? From recombinant manufacturing, we've got to look for wild type contamination because that can be really important. And this boils down to the CMC section, and I may even discuss a little bit about CMC in a few minutes. What's the DNA distribution? The shedding that's occurred from the humans and the animals, and we talked on the inflammation, but more detail on what is the immune response. Non-viral vectors, the less cytotoxicity is going to be seen, less mutagenicity, less. But you need to know your biodistribution, you need to know your formulation toxicology, how you're going to deliver it, what's the autoimmunity, all these things, all going to be important in the preclinical design. The risks you need to really consider is the test materials, process impurities, comparable to the GMP for clinical, your target organ, but is it getting into other organs, how specific is that, environmental impact, especially from genetically modified organisms, and risks that are vector specific as well need to be considered. So vector selection is critical, because you want to use a vector you can use in the clinic. You can't change it, you can, but you're going to have to repeat a lot of work if you do. The species, again, is it a homologous gene? Is it in the species? It makes it relevant. Are you going to have to use transgenic animal models? All these things need to be considered. The clinical relevance as well from your results needs to be important. You're looking for immunogenicity. It's all done on a case by case. You need to demonstrate the pharmacological relevance with that virus and that infective species. You may have to use a different vector to get your gene in there, and you need to know this from the start, because that's going to impact on how you present the data for you moving into the clinic. Protein products as well, there's functional effects in the species. We talked about biodistribution. This is so important. You need to understand this before you can get into phase 1. Again, and if it goes in the germline, it's just game over. So there's a lot of information that's going to go into this planning process, and it's a specialist project. This is not something for a generalist preclinical expert. They can probably run it and do your planning, but when you get into actual implementation, it's all about specialist expertise. Come to us, we can help you find that expertise. We have got expertise in-house, but I'm going to put another video video up, so go to video E and we'll get you some more information. Thanks.